Good morning, everyone. So, in continuation to our previous videos, in this video, I'm going to explain how to write the abstract. So, especially we discuss about writing this abstract by taking few abstracts from various journals, and these papers are published in journals like Nature Publication Group, then Springer, BMC, Elsevier. Actually, I have compiled almost 50 abstracts for explaining the structure of the abstract. By seeing the different abstracts from different publishers, we will understand the overall structure of the abstract, and we can identify some shortfalls in the abstracts of some publishers. So let us start with the first abstract. So coming to the title, so the title of this paper is WGCNE and transcriptome profiling reveal upgenes for key development stage, seed size, oil content between wild and cultivated soybean. So in this paper, they have done experiments on soybean of wild and cultivated varieties. So they tried to find out the size of the seed of this soybean and oil content that is produced from these seeds. So coming to the abstract, see here the motivation or introduction and background they have written in the first two or three lines. So they have given the introduction of this soybean. So coming to these two lines, they have written soybean is one of the most important oil crops in the world. So they are trying to explain the importance of this oil crop or the significance of this oil crop being an important oil crop in this world. So there are some important things to explore in this topic. So the domestication of wild soybean has resulted in significant changes in the seed oil content and seed size of the cultivated soybeans. So the domestication of soybean of this variety that has resulted in some changes with respect to the oil content and size of this soybean. So this is the actually background or introduction or motivation that they have written here. Next, coming to the purpose and objective from this uh, abstract. So to better understand the molecular mechanisms of seed formation and oil content accumulation, so they have used some varieties of the plants. So they took the genome database of these varieties for transcriptome analysis. Actually, research problem they did not write in this abstract. They should write the research problem like molecular mechanisms with respect to seed size, oil are unknown. So though this is a very important oil crop, so the domestication of this soybean has some changes with respect to oil content and all. So there are no studies that could explain the molecular mechanisms with respect to the seed oil content and also the size. So that research problem they did not write here. So after writing the research problem, they can write the objective, that the objective of this study is to understand the molecular mechanisms with respect to the seed size and oil. If they would have written this, so this abstract could be very well understand by everyone. So this is the object and also methods. See, so they have mentioned the method that they have used here. So objective they have mentioned, but a research problem they did not mention here. So next, results of the research paper they've written, and next, conclusion also they have written here. So conclusion is, so understanding of seed lipid metabolism and seed size during seed development. So this study helps to understand these seed lipid metabolism and seed size during the seed development, and identify potential genes responsible for this size of the seed or the quantity of oil that is produced by this seed. So this should be in the conclusion. So they have written the conclusion. So the objective is to identify the molecular mechanisms or the genes responsible, and when you see the conclusion, so they have identified that molecular mechanisms, so from this study, so the objective must be fulfilled. So the result of that objective should be written in the conclusion. So next, exploring biomarkers for autophagy mediated macrophage paroptosis in arteriosclerosis. So this is another abstract. So from this abstract we can see the purpose and objective. So they did not write the background of this arteriosclerosis, so directly they have written the objective or the purpose of this abstract. So both are same, purpose or the objective. So before writing the objective or purpose, we should write the background of the subject. So from the background we could understand the significance or the importance of the study or the research paper. Should have written that background here. Also they did not write the research problem, so that is also missing here. See, in the background they should write what is the macrophage paroptosis, and how this macrophage paroptosis is involved in arteriosclerosis. So once they write these contents, so that motivate the reader to go through the research paper and research problem also. So though this disease and this process is well established with respect to this disease, so the process of this macrophage paroptosis is well established in the arteriosclerosis. So the molecular mechanisms or the genes that are involved in this particular process is not known or not well known. Therefore this study is aim to investigate the macrophage autophagy related paroptosis arteriosclerosis. So the study tried to investigate the molecular mechanisms. See, they did not write very clearly what they're exactly investigating. So that is missing in this abstract we can see here. So after that, methodology. So they written the methodology that they have used some WGCN and GSEA or software for conducting this study, so for studying the objective. Next, results they have mentioned here. Next, we could see the conclusion. Our data identified hub genes responsible for macrophage autophagy mediated paroptosis and arteriosclerosis. So the objective is fulfilled. So their objective is to identify the molecular mechanisms of the genes responsible or involved in this particular process, and in the conclusion they have written that they discovered the genes that are involved in this particular process. Coming to the next one, so STAT1 suppresses the transcriptional activity of TRIM21 in gastric cancer. So this is the title of the paper. Coming to the abstract, so purpose of this one is this particular protein TRIM21 has ligase activity, and this TRIM21 is involved in the regulation of various biological processes in vivo, and it has been found to have a strong association with the cancer. So that is the not the purpose, so it is a background. So this is the background or the introduction. In this study we are aimed to investigate the TRIM21 role in the gastric cancer. So that becomes their purpose or their objective. So research problem, they should write that however the TRIM21 function in gastric cancers is not unknown. So that becomes the research problem. See, that is that they should write. So here see the objective they did not write. The objective of the study to find out the role of this particular TRIM21 in the gastric cancer. That should be the objective. So that becomes the purpose. Actually whatever they have written under the purpose or the background or the introduction or the significance of this one, not the purpose. So that just the motivation or the background part of the research. Okay, next let us see another abstract. So coming to this abstract, the title of this paper is a distinct pattern of gene expression changes in the colon and striatum of young mice overexpressing alpha synuclein support Parkinson's disease as a multi-system process. So they took the colon and striatum from young mice, and they found some protein overexpression of this alpha synuclein, and they are trying to see the distinct pattern of gene changes with respect to these tissues. So okay, so coming to the background, so background means that, so it is nothing but the introduction or the significance of the work, or significance of the work, or the introduction of the work, or the motivation part of the work. So after this introduction or significance or motivation or the background, so you must have the the research problem, and after research problem we find the purpose or the objective of the study. After this we find the methods used for conducting or for finding the answers or the solution for the research problem, and then the results which they obtained from the study. So after methodology results we find then a conclusion. So coming to this background, so here they are mentioning that the gut brain axis has a role in the Parkinson's disease. So the gut and brain axis has a role in the Parkinson's disease. So they're explaining the significance of this gut brain axis under the background. So also they're explaining the human wild type alpha synuclein overexpression of this human wild type alpha synuclein exhibits slow colonic transition. So in mice they were expressed alpha synuclein protein, alpha synuclein protein in mice, and when they were expressed this alpha synuclein protein in mice, so the gut and brain transition of this disease is delayed. So usually this process, the gut to the brain transition of this disease causes the motor problems in that individual, and when they overexpress this protein, that has delayed the gut to brain transition. So that is the actually they're mentioning, they're highlighting in the introduction part, but actually these authors, they did not do these overexpression studies. Just they're explaining that there is a gut and brain axis involvement, and overexpression of that particular protein delays the gut to brain disease transition. And that is the introduction. Under the introduction we could see, and next we can see under introduction, identifying the molecular changes in the gut could provide both biomarkers for early diagnosis and also gut targeted therapies to prevent progression. So background, they are mentioning that identification of the genes could helpful us use these genes as biomarkers, also to use them as a therapeutic agents also. So that means this kind of this is the research actually lacuna that is they're mentioning. So they can write even however the biomarkers for the diagnosis of this particular disease, and the genes to identify as a therapeutic agents is not very well known. So that becomes the research problem. So clearly they could write that however the molecular changes are with respect to these conditions are not well known. So that becomes the research problem. So then objective, so after research problem we could see the objective here. So identifying the molecular changes, so the objective of this study to identify the molecular changes in this particular gut brain axis. So that is their objective. And the methods, we can see what are the methods they have used to find the answers for their research problem. Then results, what are the results they have obtained after implementing these methods in their study, you can see under the results section. And conclusion, we can see whether the objective is fulfilled or not. See, and conclusion, when you see the overexpression of this particular protein disrupts gene expression in the colon, so they want to identify the changes in this particular tissues under overexpression of this particular protein. So because their objective is to know the molecular changes, that means genetical changes. So though there were studies to explain this protein overexpression delays the transition of this disease from gut to that brain. So no study attempted to find the molecular changes that happens during this transition period in the presence of the overexpression of this particular protein, because this particular protein overexpression has delayed the progression of the disease. But how this particular protein overexpression has impact over the genetic changes is not studied so far. So that's why they attempted so that's why they designed their study in this direction. So in the conclusion also, they mentioned that overexpression of this particular protein disrupts the gene expression in the colon. So when they compared the control or the normal mice with the mice which has this overexpression protein, so they took the tissues of these two mice, the control and the overexpressed protein mice, and when they compared the gene expression profile, so they found that this overexpression of the particular protein in mice has changed the genetic pattern or the gene expression. So that they mentioned here. So that means their objective is they have achieved their objective. So they identified the gene changes that come because of the overexpression of this particular protein. So next, while changes in colon gene expression are highly related to known PD relevant mechanisms, they are distinct from brain changes and in some cases opposite in direction. These findings are in line with the emerging view of PD as a multi-system disease. So when they expressed so this particular protein in a particular mice, and when they collected the tissues like gut and the brain, so they got different results. So the gene expression in the gut region and the gene expression in the brain of that mice are not same. So there are some differences. So that's why they mentioned that this is a multi-system disease rather than a single system disease. So multiple systems are involved, and multiple systems progression or multiple systems disorders in this particular condition are not same, or completely quite different, that they found from their studies. So these are some abstracts that I explained in this video. In our next videos we will explain the remaining abstracts also. So almost 50 abstracts have compiled from various journals, and by seeing all these abstracts we get a full understanding of the structure of abstract, and what must be there in the abstract what must be there in our abstract to publish our scientific results. So thank you.